master's in animal science from the University of Ilorin in Nigeria, and he also had his bachelor in animal science from the University of Modibo in Nigeria. So if you are just joining now, we are coming to the second session of this webinar, where we are, prior to this, we already have the first session where he introduced us, theoretically to the 16S analysis and all its, content. But now we are going to a practical session using the R Studio to demonstrate how you can run the 16S analysis, even the MICROECO package. So Dr. Amin, you can start.
Okay, thank you so much, Dr. Samela. Let me get started. Hold on, I'm trying to load my R Studio.
Hello.
Hello.
Yeah. Doc, can you please, are you going to share your script so that we can follow you along?
Yes.
Okay.
Yes,
Okay.
I'm sharing my screen.
The script, I mean the R script.
Okay, yes, I was asked to delay sharing of the script until the end of the session.
That will be shared at the end of the program.
Yes.
Oh, okay, then no problem.
Okay, so can you see my screen?
I think it is trying to come up.
Yes.
Yeah,
Okay.
we can see your R Studio screen.
So, good day to everyone once again. I am Abdulmumin Babamin, and I will be taking us through the second session of this presentation today, which is the practical session. So this is going to show us some of the basic downstream analysis of microbiome, 16S microbiome data set in R Studio. So I usually start my script by clearing the global environment, graphics, and to set in the working directory. So let me load the library. MicroECO is the software we'll be using today, is the most important software, although we are using so many packages, but this will be the most important package we will be seeing today. So I don't know. I have showed us how the OTU data looks like, right? So let me load my OTU, sample data, and the taxa. So we'll see how they look like. I want to create first, I need to load my OTU. If you are conversant with R, this is a CSV file. I am loading it. And this is the OTU count. This one is the sample data. And this one is the taxa data. So as you can see, they are already loaded. For the benefit of those that are not very conversant with it, they are already loaded. If I may want to show us the data structure. So this is how the OTU count looks like. These ones are the OTU, these ones are the samples. All the ones beginning with A belonging to a particular group. That is, these are replicates, 1, 2, 3, 4, 5 replicates. So this is how the data structure for the OTU looks like. For the sample, if you look at it, we have the sample information, we have the group replicates, we have the group, and we also have the management system, either the feedlot or grazing. So this grouping, so depending on the factor we'll be considering. And for the taxa, for the purpose of this presentation, I just trimmed it to the 3 most important categories we will be considering. That's the phyla, the genera, and the species. The phyla, genera, species. So these are the 3 most important ones that we'll be considering today. Although the taxa is much more than this, you have the family, you have the order, you have the kingdom, although all of them are from the same bacterial kingdom. So these ones are the ones I just streamed in the, but however you are using, you may want to, it's the same procedure. So this MICROECO software needs us to bind all these 3, that's the OTU, the sample, and the taxa, into a micro table object. So this OTU, this is the OTU, this one we created, and this one sample table, and this taxa. So they will be the ones who will be binding together to form this new object, OAAD. So now our OOAD has been successfully binded without any error. So we may want to, this function is to trim the data set and removing consistent data. So, and this is just to calculate relative abundance, that's normalization by relative abundance. So OAD, cal abundance. So this script will calculate the relative abundance. So the result is stored in the object. So we have already checked our data structure. We know how the data looks like. So now we will calculate the alpha diversity of our data set. This function calculates the alpha diversity, trans alpha, new, and the data set, we are going to put our our object, working object, that's the micro table object. This one, OAAD, and the grouping for our thing, for our alpha diversity. Is it going to be the group? That is our treatment control or the management system. So let's use the group. So alpha diversity not found, calculated, as my results showed the transform diversity is stored. So it has been stored here. So if we check the alpha diversity that was calculated, it calculates for the observed OTU, CHA1, Shannon index, Simpson, Simpson, inverted symptom, Fisher, so many of them. So many of them, but we are going to be visualizing them to see what has been done. So now we'll calculate differences among the alpha diversity. Just like I told you, differential statistics. So the alpha diversity, for us to know if there is a difference or there is richness or evenness between the sample, we need to use a statistical analysis to statistically tell. So this one is going to be using Kruskal-Wallis test, or if we want to use another, based on our data set, if we want to use another statistical this thing, we may just, you can just look at seek the help function and see the various statistical analysis available. Help trans alpha. So if we check the help, this thing, we will see various statistical analysis we can use. Cal difference. So these are the various statistical: Kruskal-Wallis, Wilcoxon, Donnet, Wilcoxon, t-test, ANOVA. So anyone. So the way it is here, you just write it here and it will be done. So the result is stored. So this shows us how the result looks like in text. So we may want to save. If we want to save, this is a function to save, to coerce back to data frame and save. So the object has been coerced to data frame and we will write as alpha diversity CSV. So it has been saved. If you want to check how it looks like, we can go back and check. So we will visualize these things so that we appreciate what we are doing. If there is anywhere you need clarification, please, my attention can be called.
Doctor, let me interrupt. I think if we go like this, it probably people might not be able to follow. So I think we should just share the data sets and the screen so that people can also be doing it and be getting the result. I think it will be better so that it will be like a really, really practical session.
Okay.
Just share
that was
it
the initial plan, really.
Pardon?
You can share it in the comment section, the data set, and also your art scripts, so that we can be running it together. Because here it might be only you, and probably someone may not figure if there's problem or not.
Yes, I understand. I understand.
So I think it's better you just share the data and the art scripts, so that people can follow. So, and from what you have done earlier, I think you have 3 data sets, right?
Yes, yes.
You can share it and then the R script so that people can follow.
So let me paste it into the group.
Yeah, so I think I agree with that. I think there was a confusion in my suggestion. My suggestion was for to share with individuals whilst you have not shared with the group. Others are experts, will be at advanced stages when they discuss the meeting.
Should I share?
Okay.
Shall I share to the comment section?
Yes, I think the comment of this Zoom program is okay.
Okay. So it has been shared to the comment section.
Okay, please, those participants that are with their computer sets, please can you download the data and then he will take us back so that we can start afresh, so we can follow.
I don't think he needs to start afresh. You just run the code up to where it stopped.
Okay,
so fine. I should start afresh?
I think it's better.
Can we give them a minute or two to
download the data? Yeah. Well, what we do now, probably we just go to the file because on there where they upload the data, because they are using their computer now. So probably they will need to edit, the file path.
Yes. So what do we do now? Shall I continue? Or wait?
I think give us like 2 minutes. Let people
Okay.
settle it. Yeah, start.
Okay, yeah.
Dr. Koja, are you also following with the?
No, I'm using different set of gadgets, so I'm not trying to get my other gadgets connected. But I'm following.
Okay.
I've done.
Because I need to. I just need to have like maybe one. But Dr. Jimo. Dr. Jimo.
Yes, please.
Yeah, please. Are you following with your with your computer set?
Yeah, I'm trying to change my working direction. I have all the data set downloaded.
No, later.
I just
Okay.
want to change my directory.
So please, I'll just be using you as the listener. From the participant, I hope everyone now have his data sets. Ready?
And I think Dr. Kakari can also help Benjamin.
Okay.
Okay.
Yeah. All right. So if they have, please can you run the data, to where he is now, because I don't think if he has done much, so we can just continue from there. Well, I mean, you can run the script where we are now, then we can continue. Doctor, I mean, I think you can come back and then continue. To make it a bit interactive, maybe you can be responding in the chat box if you are good to go.
So can I continue? Are they ready?
Can you get some response in the chat box, please? Probably they are still busy with the data. Okay, someone said already. Okay, we have 2 people already. Dr. Kari, I guess you're also following. So are you ready? Please, if you have some, because we don't know how many people are actually, with their computer sets.
Check, Smile, I check the chat box. I think Juliet is ready.
Yeah, Juliet.
Dr. Jima is also ready.
Yeah.
They are responding in the chat box, so pay attention to the chat box.
Yeah. So, okay, there's another person also. I think he said, now we are ready to continue. Yeah. Okay. Dr. Amin, please, you can continue. Others will follow. Yeah.
All right. Okay. So we have just saved the alpha diversity calculation and calculated via Kruskal-Wallis. So this is another method of calculation using ANOVA. So this is just to give us different calculation methods depending on the data set. But the Kruskal-Wallis is more appropriate for microbiome data set because of the nature of microbiome data set, non-parametric nature. So this is so we can just run this 1, 2. So after running them, it will calculate. What it does here is that it calculates differences using ANOVA. And here we calculate the matrices, CHA1, if we remember the CHA1, the other, we want to plot it here, to plot the. So I think we are beginning to see results of what we are doing. So this is the CHA1 alpha diversity matrix between the groups. So I think there is a little error here in the data set. Sorry, this is not our data set. This is another data set, OAAD. Please, all of us should take note of this data set. It's supposed to be OOAD because it's calculating it for another different. So this is our group, as we can see. I'm trying to zoom, but so let's look at this plot here. As we can see, based on the CHA1, there is difference between this treatment 1 and treatment 3.
I think you can use the zoom function. So it can be. So I think you can use the zoom function so people can see it clearer by clicking on that zoom. Yeah.
I wanted to use the zoom function, but I think
the zoom function will open another page, so it might not show.
Oh
yeah. So I have to close the scripts.
I get it. Go ahead, sir.
Okay, you can go ahead like this.
So this is it. So since we all have the scripts and we know how to differentiate significant differences, I don't need to waste much time trying to explain this. So this is just how to have our plot, CHA1 plot. And if you want to save it, we can manually save or we write it down using so this is just to add some little formatting from ggplot. So you can modify the colors the way you want it. So I'm not really a very very good in R. So I do play with it, but it's giving me headache. I'm still learning. So that one can be modified according to your preference with ggplot too. So we want to calculate beta diversity and plot the PCoA plot using ggplot2.
As you can see, Dr. Junwo is like saying going well so far. So please, if you are following, can you be indicating that so that we know that you are going with the.
Okay, I'm moving so fast.
No, not you, sir. I'm saying that the people can be showing in the comment that they are getting
Yes,
it right.
if I'm going too fast, please let me know.
Yeah, can you spend 1 or 2 minutes to? It looks like the plot we have now is different from the rest, although I seem to have an idea, but a quick interpretation of this might be helpful to everyone on the platform.
Okay. So this one is the PCoA, principal coordinate analysis that we talked about is an ordination method used to display, to show us the beta diversity, calculated based on Bray-Curtis. I will show you the calculation method based on Bray-Curtis or Jacquard, but this one was calculated based on Bray-Curtis. So these are the different groups. So from the function, it was asked to differentiate the groups with using shapes. These ones are the replicates. All the dots, they are replicates, and the shapes represent each group. So this is just to show us this TR treatment 1 group is shown by this orange color and the triangles. It shows that this particular treatment has the samples are more closely related to each other than this particular treatment 3, which has more diversity between the microbiome group in the samples. So this is just a simple explanation
Hey,
of
can
this.
you come again? I didn't get that. I'm sorry.
Just we are talking about diversity here. Diversity meaning how they differ, how the samples differ in composition of bacteria. Is it clear? So
Following. So you are trying to say one is more diverse. That's the point I want to try
yes,
to.
when it is bigger, it means the samples are more are far away from each other. These ones, the dots are not individual bacteria or taxa. They are replicates. So meaning the replicates of that particular group are more far away from have high diversity. That's why the distances are far away from each other. It was calculated based on Bray-Curtis distance.
Okay, so if I get you well, it simply implies that the bacterial composition treatment 3 seems to have the highest diversity, followed by the control.
Yes, yes, yes.
Okay.
So that's it. Simple.
Thank you.
So we can save the plot using this function, or we can save it manually. So depending on the way somebody wants to save it. And PCA, although PCA is not really appropriate for microbiome data set, the PCoA captures it all, although one may want to show the PCA, but from what I read in several texts, the PCA is not very appropriate for microbiome data set, though we are all learning. I stand to be corrected. So we can run the PCA, but that one is calculated based on Euclidean distances. It's also similar to this, but it works on the raw data. This one is more robust when it comes to PCoA, is more robust when it comes to microbiome data set. So let me just run it so that people can, it doesn't, there is no much difference. So you can see the PCA of the same data set is completely different from the PCoA, the organization of the eclipse. So This one is calculated based on Euclidean distances, unlike the other one based on the Jacquard dissimilarity matrix or Bray-Curtis. However, our own was strictly based on Bray-Curtis.
So is there different, can you please tell us possibly how to differentiate? Does the interpretation differs?
No, the interpretation is the same, but the PCA. However, the PCA applies to a lot of things, not microbiome. It's the purpose are different. The PCA is just showing like distribution, just how the sample cluster together or not. But this one identifies the microbes and try to relate them to each other, like beta diversity, try to see how samples are closely related based on individual microbial taxa. Do you understand? But PCA doesn't look at individual microbial taxa. It looks at the samples and are they close or are they diverse? It looks at numbers, but the beta diversity looks at closeness. The PCoA, that's the difference. This one is particularly suited for microbiome dataset, while the PCA is not. It applies to lots of things, lots of dataset, metabolomics and other things.
Yeah, well, I use PCA actually, but I'm new to everything microbial, so that's what I'm asking. And
Okay.
there's a
Yes.
kind of different way we interpret
Yeah,
it.
PCoA is
That's what I'm trying to
particularly
ask.
suited for microbiome data, so it shows how closely the microbes are related to each other.
All right.
So,
excuse me, please.
yeah.
Yeah, this PCA that you are talking of, PCO2.
Yes.
I think the eclipse are almost the same. And the data you have imputed, everything we have there.
Can you
hear
please
me?
be louder?
Okay, all right.
Your voice is very low. Can you speak louder, please?
Oh, okay, all right. Can you hear me now? Hello?
Yes.
Oh my.
Oh, okay, all right. What I'm saying here is the PCA and the PCO2, the graph is almost the same. The eclipse are the same in a way. And all the keys there you also have there is also the same. When you talk about closeness, you're also talking about values. That's numbers, if I may say so. Am I right?
Pardon?
Okay, I said when you talk about the closeness of the PCO2,
yes,
it has to do with values. That's numbers.
Yes.
And so this PCA2 is also about numbers.
Yes.
So I think there should be a clear distinction between these two, such that when we want to interpret, we know what we are saying.
Yeah, actually, I don't understand the question.
Yeah, I think the question he's trying to ask is that how do you try to differentiate between PCoA and PCA? And one of the things I took away from your explanation is that it has to do with the approach or methodology through which the result is generated. For example, the PCoA is generated using Bray-Curtis, but the PCA here is generated using Euclidean distance. So based on that, there's likely to be some slight differences. I was also a bit confused with this PCA in the first instance. That's why I asked, because the PCA I'm used to as a trait person is like, we project the trait, it shows us the variables, so we know which ones first on the, PC1 and PC2, which ones have, the strongest, contribution. But, this is a new field to me. So that's why, kind of asked that underlying question. So that's the question he was trying to ask. I don't know if I amplify what you are saying, sir.
Yes. Thank you.
You're welcome, sir.
Okay. Yes, based on the calculation method, they differ. However, based on explanation, they are almost similar, I would want to say. I think that's the easiest thing to say. They are similar, but if you use PCoA, what I'm saying is you don't need to use PCA again, because the PCA is more like not specific for microbiome, while the PCoA is specific for microbiome.
Yeah, I think we can move again. I think it's clear.
Okay.
Because
Okay.
when you look at although what he was saying that he's saying that everything is similar, but actually the percentages for PC1 and PC2 is different between that of the PCoA1 and
Yes,
PC.
yes.
So definitely
Okay.
it indicates that, yeah, definitely it indicates that there are differences in the calculating
Yes.
the PC and PC.
So,
I think
and okay, I want to just go back a little bit, ordination. Okay, if we look at this measure, when we are calculating the beta diversity, it shows Bray, meaning it uses Bray cortis. So if
Dr. Amin, can you be using the
yes.
line number? Can you be using the line number there?
Okay, line number 84. Okay. Line number 84. So that's where to change the measure for calculating it. One may want to use another method. So if you use the help button, you will find several methods that one can use there. So just to bring us back to that. So this is to calculate the distance between our samples. If we want to know, calculate the distance using the samples. So this is the Bray cortis distance that was used to plot the PCoA. So, and this is to show that the distance between the samples in control and treatment 1 were significantly differ from one another, while treatment control and treatment 2 also differ significantly, while others do not differ significantly from each other.
Sorry, what line of code gave you that plot?
Okay, line 128.
Okay. Trying to reproduce it.
Okay. So
1, 2,
we may want to see the clustering of the samples, because just as I said, the PCoA is an ordination method which is used to show the clustering. That's a visualization. Another clustering of this beta diversity, the one calculated via Bray cortis. So one may want to see another visualization method where it's showing how closely related they are to each other.
Sorry,
So
Doc, my own line. Sorry, Doc, my own line 128 is not giving me that that NS you have at the top. I did got the box plot, but it's not showing that
probably you missed there's something with R when you miss a line. So that line is a prerequisite for the subsequent ones. So if you missed one line, you need to be running the code as you can't jump other lines and go to 128.
It's all right. Continue,
yes.
Please.
Okay, sir. So you have to run all those lines between where you are and 128. So this is to plot the clustering to show you like a phylogenetic tree of how closely related they are, the samples in the grouping based on Bray cortis distance. So this is a palm ANOVA, which is a permutational ANOVA used generally to see how the taxa differs from one group to the other as a whole So just to see, is there any difference in the taxa regardless of species or just the whole taxa as a whole? So one can decide to run it, and the result is saved. You can write the result and see. So this is the result. So the whole taxa as a whole showing a p-value of about 0.47, not significant from per maneuver. But when we are running another specific test for microbiome like LEfSe, that one will look at individual species and will also have other indexes apart from, which is different from that of per maneuver. So this is the GG3 and we have already calculated the relative abundance. We want to now see how we can draw a Venn diagram of the samples to show us how which ones are having similar samples or similar taxas and which ones are having different OTUs. So this place should be changed to, okay, yeah, it's OOD, it's okay, no need. So this is a Venn diagram. Shows the number of similar OTUs cutting across all of them. So this is to show 4,808, I think, cut across all the 4 samples. While unique ones are 6,200 for this, 6,200 for this, treatment 1 having the lowest 2,500, this one 5,000. So this is just a way to interpret this.
Dr. Amin.
Yes?
Sorry, I don't know if Smiler is here. Somebody should be helping you to attend to questions in the chat box. I think there are a lot of questions.
Dr. Samela.
Yeah, actually I'm here, but this is the same question that he has explained, earlier on.
Okay. Please be responding to them, even if it has been explained. Tell them that it has been explained so that at least they can refer to the video later.
Yeah, but maybe you can just quickly, come again on the question. She is, because she saw that there's a difference in her PCoA and PCA. So please, she wants to know what is exactly the difference. That's the question.
So this is to calculate LEfSe for all the taxa level data set OAU calculation method, differential analysis, just to calculate LEfSe using group as a factor at alpha level 0.05 and taxa level, all the taxa levels, and filtration threshold as the taxa to use for this should have abundance of relative abundance of at least 0.01%. That's times 100. So we are trying to do the LEfSe plot to calculate differential abundance. So this is a calculation of differential abundance for this group. It has been okay.
I think we can reduce the font size to make the labels a bit clearer on the y-axis.
Yes, yes. Okay, I think I need to use ggplot for that. This one will give me the, will waste our time. Let's just proceed. I'm not very conversant with the use of ggplot. So let's manage it this way, please. As I said, I'm still a learner with regards to R, though I have used it for about 3 years, but R never ceased to amaze me. So this is just to show the differential abundance between them, though no statistical test to, no specific statistical test was carried out, but just to show you the abundance in different groups. Like these ones having the highest abundance, these ones having the lowest abundance in all the groups, and this is the relative abundance. But we limit it to the 1 to 200 taxa, to bring 1 to 200. So it doesn't really say anything here until we get to the LEfSe. So this is just a cladogram of the LEfSe. So this is really a very difficult thing for me to interpret. Dr. Samela, you have to come in here. Cladogram, I've not used it. So I have been plotting it, but interpreting cladogram has always been very difficult for me. So we may want to go and check the interpretation. I don't really understand the cladogram, though I have checked several times. It's giving me a headache to interpret, please.
Probably, I think Dr. Kojo, because he's into, or Dr. Kari, because they are into genetics, they normally use, because this one has to do with the,
Yeah.
just like.
So I'm more interested in there are all those visualization methods, depending on your area or specialty, which one you want to use or not. So I am set to plot the LEfSe, so I need as much colors as possible to help differentiate. So I'm using RBrewer to collect all the colors from palette 1, palette 2, palette 3, palette 4. So combining them into one big palette. So I need to this is just to combine colors so that when it comes to the abundance, it will be easy to differentiate one taxa and the other from all these color set. So I am calculating the abundance, the relative abundance for the 30 highest taxa, number of taxa 30, that's the top 30 taxa, using group as a factor, and it should show abundance of 0.01, that's 0.01%. Any taxa having lower than 0.01% will not be captured here. So, and the taxa rank for the abundance is genus. So this has to match specifically to our taxa file, the way it's written, genera or genus, whichever one, it should capture that.
There's an explanation provided by Dr. Kojo on the.
Okay.
Yeah, in the chat box, so people can just read it.
All right. Okay.
You can continue.
So, Is this plot going to?
Maybe you can increase that panel. When you
Okay.
when you click on the environment, I think then it can go up a bit.
Okay, I should just decrease the panel or increase.
Yeah, you can increase it so that we can see it from there.
Okay. Can you see this page now?
We cannot see the page because if you use Zoom, we cannot see it. But probably
Oh,
you can drag it up and also the other
okay,
side.
I understand.
So,
Okay.
You can also use the viewer so that we can view it in the complete mode, and then later on you come back.
Oh, I understand. Okay.
the problem is if you use viewer or Zoom, it will open another page. So he will have to share that page.
I see.
Oh, you have not been seeing the plots I've been opening here because I only shared the Zoom page.
Yeah.
Okay. Let me try and so, but since we all have the codes for those running.
Oh, can you just copy it and put it in the chat box? Probably you can see it.
All right. So let me just save it first and then I will now paste in the chat box. Okay. So I think I dropped it in the chat box. I'll just be dropping them in the chat box. So can we all see it?
Yeah, I think it's coming up.
Yeah. I can see it from my end.
Is anyone able to reproduce it?
I think Dr. Jimo, Juliet, Benjamin, I don't know. Have we been
So
able
this
to
is just a line chart.
No, I'm trying to struggle with GG3 package. I'm still unable to install it. It's not available in Crown or something.
Which one?
Okay, if it's not available in CRAN,
It's giving me some kind of error.
Which one? Which package?
GG3. GG3.
Oh, GG3 is actually not direct, it's a proxy package.
Yeah, I'm trying.
I will send you the.
I'm trying to troubleshoot to see how to get across that.
You use Bioc Manager package.
Bio Manager, yeah, I just checked that out. Thanks, man. Yeah, I'm trying to find a way around it now.
So this is just a line chart to show us the abundance of top 5 taxa, so we can modify according to our preference. So this is a heat map of top 40 taxa at phylum level. And so let me share that as well.
Yes. Yeah.
So this is relative abundance for top 50 phylum. If we want more than that, we can change it here, but the space cannot take much. We can write, save the relative abundance, and here we can plot it, format the plot according to our, and save. So let me share the relative abundance too.
Dr. Amin,
Yes.
how is this different from the previous one? Because I can see they are all estimating the relative abundance of the bacteria community.
Okay. Which one is the previous one?
The previous one, this one, I think.
Is it the line plot or?
Yeah, I think it's the bar graph.
Oh, the bar graph. Okay.
And then
One is
the heat map.
okay. Yes. One is for genera, while one is for phyla. The second one, this is for phylum. The other one is for genus.
So the bar graph is for the phyla, and the
No,
heat.
both are bar graphs. I think you are referring to the one we produce using code 246, line 246, right?
Yeah.
Yes. It's a bar graph of highest 30 most abundant genus at genus level, while this one is a bar graph of 50 most abundant phylum. So they are all the same, but for different levels.
Sure. Okay. All right. Thank you.
All right. So, and this is the LEfSe plot. Even the LEfSe linear linear discriminant analysis effect size can be done at genus level, at phylum level, at species level, depending on which one. But in most publications I have seen, they are mostly calculated as genera and at phylum level for greater capture. So this is still using. So if you look at the bottom here, start Kruskal-Wallis rank sum test for the group. 21 taxa found significant. After p-value adjustment, 21 found significant. Minimum LDA score 1.42. Maximum LDA score 3. So based on this one, you may want to peg. So if you want to plot your LEfSe now, LEfSe, so you have to peg the score at least, it should not be more than 3. Because if it is more than 3, then it will not capture anything. If you put 3.5 now, you can see the maximum score is 3.39. So if you have an LDA score as a threshold 4, then it will not capture anything. when plotting LEfSe. So this is just calculating LEfSe, but you will be the one to peg the threshold for significance. In most articles I've seen, usually people peg from 2 to 2.5 to 3, depending on how many taxa you have seen to differ significantly based on that. Because if I have seen a lot of taxa, like if this maximum was like 5, I can decide to say, okay, let me peg 4 to have more screening, to be more accurate. But since it is 3, I can decide to peg 2.5 or 3. Maybe it's just one taxa that was even above 3. So it's only when you plot that you will see. Or you can decide to save this. Okay, if I save this, I need to coerce to data frame, then save. So now if I save this and I check, I will see the taxa. So it's just that even if I check now the screen, I will need to reshare it. So if you get to this place, you will save and check. Or let me share the file then. Let me share the file, then we can check. So this is the result for genus LEfSe. So we can decide to use ANOVA. This is just to give us options of what we may want to use. So if we want to plot the bar plots for LEfSe, you can see the threshold here in line 343, 2.5. So we want to change it. If I want to put 3 now, I will put 3 and we'll see how many we are plotted.
So
Let's use color viewer.
I mean, you mean you can use ANOVA for the
Pardon?
You mean you can use ANOVA for the abundance also?
No. You can use ANOVA just like you use in any analysis to see
the
differences.
difference.
Yes,
Okay,
but the LEfSe plot, the LEfSe plot, you use your LEfSe calculation, LDA, and Kruskal-Wallis test to do that.
What I'm trying to say, if you use ANOVA, can you also
Plot LEfSe?
No, can you also plot the ANOVA table? Like the difference?
Yes, you can plot the abundance, the differential abundance for ANOVA. You can use box plot or anything you want to use, but
okay.
Microbiologists have really frowned against using ANOVA on microbiome data. So that was when this LEfSe was developed by Harvard-based scientist. Is it Curtis Hunt over something like that? His name. So the Harvard-based scientists developed this LEfSe, and they believe it has more statistical robustness to identify differences in microbiome data, which ANOVA was not specifically designed for microbiome data. So LEfSe, ANCOVA, ANCOM, we have lots of them that were specifically designed for microbial data set.
Okay, thank you so much for the explanation.
So this is the LEfSe plot. Let me save it and share. So if we look at the LEfSe plot that I shared in the comment, can we check the LEfSe plot I shared in the comment? I think this addresses the question raised earlier when I was presenting the PowerPoint. Somebody confused with the negative. So we see this software is giving us, I think that software is a Microbiome one package that I used, but this one, MicroECO, gives all of them a positive value. So, and if we look at the plot, we will see that very few, about 1, 2, 3, 4, 5, only 5 we are able to have up to 3 LDA score. So all the rest are between 2.5 to 3. So if we had put 3, it will capture only 5. So one can just, at your discretion, you look at literature and you put your threshold based on the results you are able to get. If my results, all of them are less than 2.5, I may decide to put my threshold as 2. Then I will capture it in the methodology that my threshold was 2. Even if they are less than 2, I can decide to put 1.5. I've seen literature or I've seen article where 1 was used as threshold. So your data will define that. The result
Okay,
you
Dr.
get.
Amin, I have a question, please.
Okay.
This figure, did you use is it the genus?
Pardon?
This LEfSe, is it for the phylum or genus?
This is for the genus at general level. Let's go back to this.
Okay, I'm coming because I just want to know. I have a question because I saw from the x-axis. Is this with the y-axis?
Okay.
One of the bars doesn't have name.
Yes, is because general it was unclassified. Yes, it was unclassified at general level.
Yeah, so I have a question here. Actually, I have a particular microbial data that when I run the analysis, I found that then when I check the data, I realized there are several, OTUs that have not been, identified at the genus level. And they form maybe a very larger part that you can see even here at LEfSe, it means they are significant. So is it okay, if you do some data cleaning and just remove those that were not identified? Or what is your suggestions there? Because you cannot publish something like that you just put it on. Because
I understand.
Should
What I do in such.
Huh?
Can I respond?
Yes, sir.
Okay. So what I usually do is in such cases, you need to clean up your data. Well, cleaning up, I don't mean you should delete them. But if you look at the way taxa are being arranged from kingdom to phylum to family or to order to family to genus to species. So for every one of it, if, for example, the phylum is available, you look at the closest that was available. If the genus now is not available, but the family is available, is it clear?
Yeah,
If the genus is not available and the family is available, then it will be an unclassified. If, for example, let's say the family is let's say lactobacillus, for example, I'm just using it as an example, then it will be unclassified _ lactobacillus. Do you understand? The closest to it.
I understand. I understand.
So,
I think that is
yes.
fair enough.
And if that one is not available, you use unclassified to the nearest you can get in the 3. So, and I have to do that for all my data sets. Sometimes it will take me a day or 2, though you use a lot of, though there are methods, there are softwares, packages that I have used. Every package, I have used about 7, 8 packages for this microbiome analysis, but this one is so far the most user-friendly I have seen. There are several packages that can take care of that. It will help you to relate it to the closest relative.
Okay, like which package? So that if I have that kind of data, I can use that.
Oh, I cannot specifically remember the package now, but if you want the list of packages, I can drop them after this. I have to compile it because it's been long that I've used those packages, but I have a long list of packages I've been using since 2019.
Okay, thank you.
But this one is the most user-friendly I have seen so far, honestly.
All right, let's continue.
So this one is at genus level, and the LEfSe at phylum level, we will calculate the LEfSe from line 363 at phylum level. And you can see that only 2 taxa, even after Kruskal-Wallis test, only 2 taxa were found significant. After p-value adjustment, 2 taxa were significant. And the minimum LDS score 2.4, and the maximum is 2.6. So if we put 3 as threshold here, we will not find anything. So only 2 of them, meaning one is 2.6 and one is 2.4. So we'll have to look at this. So when putting threshold, sometimes in some articles I publish, I put threshold for genus separate from threshold for phylum. Because you may find if you put a lower threshold in some cases for phylum, for genus, you find too many significant taxa, sometimes even more than 50. So for you to start giving explanation for them in an article is difficult. So it's better to increase the threshold so that it will focus on the most significant ones that have greater effect. So, but at phylum, if you use that same threshold, then you find it, you find out that very few phyla could be able to cross. So you have to reduce the threshold for phyla in such cases and leave the one for general. So this is to plot phylum LEfSe. So I use 2.5 as threshold, the same one I used for the general, and only one phyla was identified out of the 2, because one is less than 2.4. Remember, one is about 2.4, which is less than 2.5. So only one was identified to be significant. Let me see. Let me post the LEfSe into the group. So as we can see, only one phyla was found to be significant. And in such cases, we may want to leave it since we already have one. But if you want it to capture both phyla, then you will have to reduce your threshold. And I think this brings us to the end of the differential analysis and the end of my practical presentation for today. If there is any question, you can write it in the comment section or you ask, please. Thank you. Dr. Samela.
Yes, sir. Thank you. Thank you. Thank you very much, sir. I think we have done well. You took us through the alpha and beta diversity and some of the things that we can actually visualize to interpret our microbial data. 16S microbial data to be precise. So I don't know if we have some questions from the audience, or if we don't know if someone need any clarification on what you have done so far. So if there's questions, someone can you can raise your hand, and then or you can put it in the comment section. And also this session will be shared in the on the OAD account, and all the related data also will be later shared. Okay, I think Ben have a question. Ben.
Yes, no, thank you very much for sharing this useful guide with us. But I would like to take us a step further before this presentation. After you obtain your sequencing, how do you blast? How do you obtain, like just a theoretical maybe guide? How do you obtain the output to know that it is maybe it shares maybe 60% with a particular phyla or whatever? I don't know whether it's just a simply by blasting on NCBI, or there are other tools that may be used in the upstream analysis before today's training.
If I understood the question correctly, you wanted to know after the sequences are obtained, right? So you wanted to know how the taxa is obtained. Am I correct?
Exactly.
Okay. So those ones are usually done at the upstream analysis, and in my own case, it was done by the sequencing laboratory using QIIME. That's why I say it has to be converted to OTU. That is a QIIME or QIIME software, QIIME, QIIME 1 or QIIME 2. It runs on this what do you call this other, not Windows, the other one. The other
Linux
program. Yes. Or if you use Windows, you need to use a VirtualBox. It doesn't run on. Yes, or you will use a Dada 2 to give you ASV, or you will use a QIIME 1 or 2 to give you OTU. And it works with this database, the Green Genes database, which is a database for 16S rRNA sequences and bacterial taxonomy, or Silver database. So that's where it's done, and it's done using that QIIME software. So that's the upstream analysis, and after that, when you obtain that sequences, that's when you will be able to use R to do the downstream analysis. You have your OTU, you have your taxa, you have your sequences. And you will be able to use R to predict the functions. That a completely different analysis on its own, functional prediction, to predict the functions of this identified bacteria. That's another different software, another different package.
Thank you.
Okay, thank you so much, Dr. Benjamin. Do we have any other, question? I don't know. I saw Juliet has been following, I think, throughout the presentation. I don't know if she has a comment or something. I think we don't have any much question here. But
hello.
Maybe
hello.
Okay.
Yes.
So I'm Yusuf. I'm Dr. Yusuf Abdulhamid. Hello.
Right.
Thank you very much, Mr. Presenter. So I just came late, very late. So my brief question is that, please, if I want to determine the absolute abundance from 16S sequencing, how should I do to maybe like using sequencing that was done using spike-in sequencing for the bacteria prokaryotic, how to determine the absolute abundance, not relative abundance, but absolute abundance. Maybe when after conducting 16S sequencing full length with spike-in. Okay.
What's the name of, what did you say? Is it the name of the sequencing platform?
Yes. It was full length, but spike-in. SPIKE, spike-in sequencing. So I have done it, and now I want to determine absolute abundance. I have already determined relative abundance of the bacteria, but now my boss is telling me to do the absolute abundance. And normally the one I have already done before is using qPCR, which I can determine gene copy number, maybe using the DNA. I have done this one. But later, when my metagenomic sequencing came back, he said, "This what we done. We have done full length sequencing using spike-in." So this is just came like as new to me, and then I searched through literature. I saw that a spike in some, they have calculated this relative absolute abundance, but it still confused me. So your lecture gave me a lot, much more insight, even though I came late. But I just feel like to ask this question, please, regarding the absolute abundance.
Okay. Thank you so much for the question. Though I am just hearing the word this thing spiking or this thing for the first time. And what I just want to add here is the absolute abundance may be difficult to determine actually. Apart from using QPR, it may be difficult to determine at bioinformatic level because it's actual concentration rather than a relative. So even in, let me just relate this question to, for example, our volatile fatty acids, where we determine actual concentration rather than percentage concentration. So I was advised, I think, by a reviewer that it's actually not entirely possible to know the actual concentration. Rather, I should stick to the relative concentration. That is why in most cases, microbial data set, you see them in relative, or they need to be converted or normalized to log base. But from qPCR, you can do that, and I'm not very conversant with that.
Yes. Okay. Yes. All right, sir. So thank you very much, sir. I have already done the qPCR, but
Okay.
maybe later I will still keep searching how to, or later I can have your contact after your maybe later. I know you can still help me on it. I'm interested. I know you can share your contacts. Thank you very much, Doctor. We really appreciate.
My email address will be shared at the end of the session.
Oh, okay. Thank you very much, sir. Thank you, sir.
You're welcome. The pleasure is mine.
All right. Do we have another question from the audience? Okay, if we don't, actually, I wanted to ask you two questions, but I think what he asked also form part of the question, because I wanted to say that recently now what they do is that they don't just do the V3 to V4, right? For the 16S. For the 16S, as you have explained in the presentation earlier, they use the V3 and V4, right? V3 and V. So now, mostly if you send for 16S, those sequencing companies, they will tell you they can do the full length. You understand?
Full length sequencing.
Yes. Mostly now, if you send them for 16S, they will say they can do the full length. If you opt, if you opt for the full length, because mostly when we do that, the V3, V4, what you eventually will see, you have a lot of species that cannot be identified.
Yes.
But if you do full length, you can relatively get a lot of species that actually most of the species will be identified, unlike when you use that V3, V4. So I think that's actually what I wanted to ask you. But with what you say, probably is because they have done the full length. That's why I think they want to see the absolute abundance. So because you said they have done the full length, actually it was the same.
Yes, thank you for the addition, Doctor. And I think you should be more updated than me. The last time I worked on sequencing data was 2 years ago, and you are still on it. So thank you for the update. I think the reason why people go for V3 to V4 region is to reduce cost. Actually, if one could do it, it's not only 16 full length 16S. One can go for the whole genome sequencing, shotgun sequencing. So it's just the cost that people are trying to reduce, that they will just focus on V3 to V4. But the full length sequencing, I think it will give more information.
Exactly.
Yeah.
Okay. I think if we don't have any question, it's better we come to the end.
Hello. I have one question, Doctor.
All right.
Thank you very much. This presentation is really, really good. I'm actually a plant scientist, but I work on disease, and the disease is bacteria related, so it's kind of very interesting. I have one question to ask. So, like the difference that we get, what parameter exactly do we look at to distinguish one species or one species of bacteria from another? Or more or less, I am particularly interested, since we are trying to look at these differences against a particular database. In what instance do we, maybe if the bacteria we are investigating is not in line or is different from the ones we have in the database, at what instance do we end up saying maybe it's a new bacteria strain, a novel one, or it's not just in that particular database? At what instance do we get that?
I think that's a very interesting question. I understand your question very well. So at what instance do we say, okay, we have discovered a novel bacteria? Yeah. Honestly, I cannot, I do not know this. I do not know how to start answering. Yeah, I have no idea actually. So, but it's an interesting question that I have asked myself this question during my own. At what instance do I know that I have identified a novel bacteria? So probably you may need to go and search, and if there's anyone that is willing to share this information with us, it's welcome. So I have asked myself this question once, but I didn't really search for it.
I think
okay, sorry.
Okay,
Yeah,
Ben, go ahead.
Yes, so if you want to probably confirm that you have identified a novel bacteria or strain or whatever, this may bring in some maybe genetics where there may be a need to sequence, and at the end of the day, you'll be in position to blast and then compare if maybe the similarity between yours and those earlier published is, let's say, insignificant, then you can then do other step of analysis to characterize them and then maybe release it as a new bacteria strain that probably you have identified as a result of your study or from your environment. Thank you.
Okay, thank you very much, Dr. Ben.
Thank you
like
for
we want to say something. Okay.
Hello. Please,
Dr.
can
Kojo?
I add a little? Thank you.
Yeah, I'm coming. Wait, please. Can you wait, Dr. Kojo, please? What? Oh, okay, Dr. Kojo, please. You also want to say something?
Yeah, so I think I would like to add to what my colleagues have said, especially the last speaker, which is Benjamin. We have worked on few projects on this. So you don't need to be an animal scientist to understand microbiome analysis. Microbiome is done even in life sciences. They can take your saliva and profile the microbiome within your immune system and be able to determine which bacteria are useful to your immune boosting or which bacteria are inimical to your health and all that. So I've heard a lot of people say, I'm in plant science, but this is not animal science. It's not animal science specific. I'm in plant science, but I still have some knowledge, basic understanding when I'm coming. Some basic understanding of microbiome analysis. So it's something that cuts across all life sciences. That is the first point I want to make. The second one, to respond to Barry's question, I think it's a very good question. And I think it's one of the core objectives of doing such analysis, to identify if there are novel bacteria or microorganisms that have been induced or repressed as a result of your treatment, of which you are analyzing your samples. And so if you do the phylogenetic analysis, like the LEfSe, the cradogram analysis that we did, they are all phylogenetic analysis to be able to identify new species or novel species that are clustering together or that are outcropped. You saw in the cradogram, there were outgroups. So the outgroups mostly are bacteria that are unique on their own, that do not share much similarity in terms of their characteristics with the existing bacteria that have been clustered together within your cradogram or your LEfSe analysis. And in that case, you need to do step further analysis, just like my colleagues mentioned. And then you can send those samples for sequencing. And based on the sequences, we can blast them either in NCBI or on any database to know the specific differences in terms of their protein structure and all that. And then based on that, you could do further analysis and release it as new bacteria. So that could be useful to your study or useful to the objective of which you conducted the study. So I think these are some of the ways you could identify novel species or novel bacteria or microorganisms in your samples. So basically it's the cladogram and the LEfSe. They can also do other phylogenetic analysis to be able to pinpoint that specifics at the species level, or even at the genus level or at the phylum level as well. Thank you.
Okay, thank you very much. Is it Umar or something? The iPhone.
Okay, thank you. Hello.
Yeah.
Please, can I add little?
Please, subsequently, it's better you use your name, please, when you are joining this panel.
Okay, sorry. Yeah. Well, my name is Dr. Yusuf Ablamit. I'm postdoc from Jinshi province.
All right, you can go ahead.
Okay, all right. With regards to what Dr. added, apart from bioinformatics, after comparing with reference genomes using tools like or maybe NCBI, so we can still use phenotypic characterization in which they were isolated. You can look at the morphology, physiology, and biochemical properties. Then you compare with other maybe species that you extract isolated or online morphology that is there available. But this is not accurate. You can use DNA hybridization, DDH, in such a way that you can perform DDH to assess the genomic similarity to know the species. If it is below the threshold of typically 70%, then it may indicate a new species. But this is what we did. We have recently identified one species. We call it Bucalderia strain B23. I'm working on it currently. It's a new species of bacteria. I isolated it from Strauss tree. Strauss, that is lemon, orange tree in Yangzhou University. So right now I'm working with it in one of the center, China, America. So Strauss Research Center here in Jinshi province. So we found new species through by confirmation after genomic references. Then later we goes to this DDH, that is DNA hybridization method. That is what we. And then you have to contact some experts after you even confirm with reference genes from NCBI. Then you can maybe run for this hybridization. Then also you have to seek for those that have previously maybe identified new species. At that instances, then you can even deposit your new sequencing into the NCBI. Then any other person came, then you are the first person that originated the finding of the species. You can even be able to name it. Okay, thank you very much for your time and listening. Okay.
Okay, thank you very much
for the addition, Doctor.
For the contribution.
Thank you very much, all.
I want to add something, please. Actually, the 16S analysis is basically community analysis, not a single, bacterial analysis. But you can also use this data to identify, some core bacteria in your. For instance, when you do all the, abundance and all this, you will see that you may get to some species that are actually unidentified. It will even tell you that this is unidentified species, because this one you also run it. When they do the sequencing, what they do, they run the OTU and everything. They will just pick those that are identified. Those that are not identified could be the one of interest for you. So if you really need it, you still need to culture that your sample. From there, you identify which kind, from the genus, maybe from any available identification, either the genome or phylum, you will know the type of bacteria it is. Then you can culture that type of bacteria before you can send for single sequences. You can take all the different colonies, and then you can send them for sequencing. This is almost what we do in our lab, because I work on lactic acid bacteria. We isolate so many bacteria. What we do, we just pick them from the fermentation media, and then we put them on the plate. We take different colonies that, as someone said, that you look at the morphological characteristics. Then based on that, you choose different colonies, and we send them for sequencing. After sequencing, then you come, you blast. After blasting, then you can identify. But for us, because what we do is just like we are mining lactic acid bacteria, so we are not so specific. We'll just go and find which one is which. Then after that, when we identify them, we come, we look at all their characteristics, and we see which one is good for our intention. Then we patent so many and we have deposited we patent so many lactic acid bacteria, and we have deposited their sequences in NCBL. But for this our webinar today is actually for the community analysis. We are trying to understand, the bacterial community using the 16S analysis. So it will be very difficult to say how you can single out one bacteria from that kind of sample. Because if you need, you just have to take each colony that is morphologically different from each other. Then you can do the sequencing by that, and then you can know which one is different from which one. I don't know if this gives you more information.
Yes, yeah. I think it's quite educated. Thank you all for the relevant information. I appreciate it.
So I think with this, we should come to the end of the program, because we actually far away from the scheduled time. I don't know, Dr. Amin, do you have anything to say, before we go to the closing remark?
Okay, thank you so much, OAAD, for the opportunity to share the little I know with everyone. It's been a pleasure having me here, and I really learned a lot from the contribution from various audiences. So thank you so much. I appreciate it.
Okay, you're welcome. Can you please share your email in the comment section, so that if there are people that want to contact
Okay.
you?
Let me share my email address in the comment section.
All right, thank you. And also, if you are also in the OAD group, he's also in the OAD group, you can ask any question regarding this or any further information. I think with that, I will call on Dr. Kojo to give us a closing remark for this program. Dr. Kojo, please.
Hello, thank you very much, Smiler and Victoria, for moderating this section. I would like to use the opportunity to thank our facilitator, Dr. Amin. In the midst of his busy schedule, he still volunteered to lead us on this. I hope all of us were able to learn one or two from this section. It might not be immediately useful to your area of specialization or interest, but in future, this may come in handy. Knowledge is not useless. We can always transfer the knowledge. So we'd like to thank all of you for joining us, and on this journey, even as we explore this fascinating world of 16 RNA microbiome analysis. Like I said, it's not only restricted to animal science, and we hope that this section has deepened our understanding and also inspiring ideas for our research areas. I would also like us to remember that microbiome holds endless potential, and your work contributes to unlocking their mysteries, especially to food security, to global health, and also to our well-being as humanity. Organisms are our friends. You might not see them with your eyes, but there are some useful that are essential for your survival and my survival. And so the more we continue to uncover their usefulness and also understand their community structure, we'll learn how to be able to adapt to them and make our living more fascinating. I would like to encourage all of us to stay curious and keep analyzing, and we look forward to connecting with you again in our next section. I wish those of you who are in China and out in that part of the world quite late, fruitful and a restful night. And those of us who have just started a great day. Thank you very much for this section.
Thank you very much, sir, for this closing remark. For those that are asking of the video of the session, actually, all OAD webinars are being deposited in the OAD accounts. And if you are in one of the group of the OAD, we normally share the link where you can go and watch not only this webinar and also the previous one conducted by OAD. And also you can stay tuned in our various channels for our next program, which could be another topic entirely. So thank you very much for being with us. But traditionally, we normally used to do a group picture. So I don't know if we can just on our videos. Dr. Amin, can you stop sharing so that we can just take a group picture just for
Okay.
videos.
Okay.
So normally do it traditionally. Can you for me, Dr. Kojo, can you help me because I normally used to have problem with my computer on this.
Let me on my camera.
Okay. Okay, so many people didn't on their camera. Please, can we?
Smiler, I think my gadget, I can't take the photo, so if you can do that from your end.
Okay, okay. It is only 2 pages. Let me
think
try. I think yeah, since it is only few people that actually open their camera, so the pictures of those that open their camera has been captured. But let me take it once again, please. So please wait a minute. I'm still trying to take another one.
Okay, I think I've been able to take 2 so far.
Okay. Yeah, also
Yeah.
I also
Okay, so let's do the last one. If your camera is not on, unfortunately, my background is a bit dark, so I cannot turn on my camera. I think my picture.
okay.
Yeah.
That's fine. At least
Okay, everybody give me a smile.
I got you already shared in the group. So thank you. Thank everyone. We can all. Okay, let me stop recording then.
Okay, so now the last one. Thank you all. Let's connect again in our next section. You can reach out to any of us if you have projects that you want us to collaborate on or to assist you to gain more clarity and be able to implement your project. Please reach out to us. We will share PPT.